Page we have 898, and the topic which we are going to study, that will be the nerve impulse, how the nerve impulse are producing. The nerve's impulse. I have already shared the screen with you. Okay. What is impulse? Who can tell me what is nerve impulse? Yesterday I told you in the previous lesson what is nerve's impulse. The message which is produced by the nervous system is called nerve's impulse, right? The signals which is produced by the nervous system is called nerve's impulse. Now the topic which we are going to take, how does a nerve impulse is going to produce or are going to start. An impulse began when a neuron is stimulated by another neuron or by the environment. So impulse can be produced in two ways, right? Either by another neuron or either by the environment, internal environment or external environment. Stimulation should be by any source, right? If the stimulation will occur from outside, from the sense organ, so we have 5 senses, right? From any one of them, or if it's from inside the body, so from inside the body, it should be physical, it should be chemical, or it should be neurological, right? Mean from the nerves, from other nerves. So it can produce in different ways. And nerve impulse can be produced by two ways. By internal, mean by neurons or inside the body, or by external, mean from the environment. Now, nerves impulse are a bit like the flow of electric currents through a wires. Nerves impulse are actually what? It is like a current or a message, right? Or a signals which is produced in the nervous system. Before going to study impulse production, we need how to rest or the resting of neuron, right? Resting the neuron. Neurons have a charge or electric potential across their cell membrane. If you look to the neuron cell, we have studied yesterday, the neuron cell, right? A unit of the nervous system. Its entire body, its entire structure we studied. Now, if you look here, it has a wall, right? So if you look to the cell membrane or the wall of the neuron, so their boundary or the cell membrane of the neurons, it has an electric potential on either side, right? They have an electric potential. The inside of the neuron has an voltage of 70 minus 70 millivolt, or it can be represented, the unit should be MV. Compared to the outside, the difference is known as resting potential. The insides of the neuron has an voltage of minus 70 millivolt compared to the outside, right? So let's suppose if you look to outside, so outside will have one value and inside will have minus 70 as compared to the outside. So the difference is known as resting voltage. The difference between the outer voltage, the outer electric potential of the cell membrane, of the cell of neuron and inside. So the difference between the inside and outside is called resting potential. Clear? The resting neuron active transport protein pumps sodium ions, which have a chemical formula of Na plus, out of the cell, and potassium ions, which have a chemical formula K plus, into it. So if you look to the active transport protein pump, right? Or we can say in simple word, protein pump, right? Protein bridge, right? So the active transport protein pump sodium ions out of the cell and potassium ions into the cell. Active transport means that the protein pump or the protein bridge which is present in the cell membrane of the neuron cell. So active transport means it's need energy, right? When it is passive pump, so it do not need energy, right? But it is active transport, so active transports, it's required always energy. So by the consumption of energy, the active transport protein pump, the active transport protein, it pump sodium ions out from the neuron and take in potassium ion, right? So sodium ion are positive and potassium ions are also positive. So it take sodium and potassium ions across the membrane, right? If you look in the diagram down right on page 898, or you can see even here, right? The gray one represents sodium ion, the gray dots or the gray represents what? The gray color represents sodium, while the other color represents what? It represents potassium ion. So if you look to the concentration of potassium ion outside the cell, it's more. But if you look at the concentration of sodium ion, so it is more inside the cell. So the one which have more concentration inside will be given out, and the one which have more concentration outside, it will come inside. Right? No, opposite to this, right? Because if it is against the concentration, so it required energy, and it is against the concentration, right? You can see here. Ungated potassium channels, protein make it easier for potassium ions than for sodium ions to diffuse back across the membrane, right? There are ungated potassium channels, which make it easier for potassium ions than for the sodium ion to diffuse back across the membrane. So it cannot diffuse back easily from the cell membrane, right? You can see here, right? This is the cell membrane. As I told you that the cell membrane in the animal cell is always be made from what? From lipid bilayer, right? So if you look, these are what? These membrane are lipid bilayer, right? But beside this, here we have embedded protein, and embedded proteins are these, right? Sodium channel, ion channels, which we call ion channels, right? So it is made from protein. This one, if you look, the first one, gray one, it is sodium channel, right? It's made of protein. Another we have the other one which can work both for sodium and for potassium, and another we have for potassium separate, right? So this is the entire structure of the neuron cell membrane, which is made from what? Which is made from lipid bilayers. These two lipid. This is also a lipid layer, and this is also a lipid layer. When both of them combines, so bi mean two, lipid bi. The lipid of protein, the lipid of the neuron, which is made from double layer of the lipid. So that's why we got it lipid bilayer. Because there is a high concentration of potassium ions inside the cell as a result of active transport, there is a net moment of positively charged potassium ion out of the cell. If you look in the cell, so there is a high concentration of potassium ion inside the cell, right, which do not require for the cell. Right? If you look here, this is inside the cell, and this is outside the cell. So inside the cell, if you look, you can indicate from the colors, we have more number of potassium ion, right? So if we have more number of potassium ions, it need to be moved out from the cell by means of active transport. So active transport is what? Active transport is that transport which required a little bit amount of energy. As a result, the inside become negatively charged compared to the outside, producing the resting potential. So if you look in the result of the inside, the result of this, the inside become negatively charged compared to the outside, producing the resulting potential. So if the inside become negatively charged and you are to the opposite compared to the outside, so it will produce what? It will produce resulting resting potential. You get the point? If you have any doubt in this, you can ask me now. If it is clear, so tell me, I can go ahead. Okay, so no question, then I will move to the next point, which is the moving impulses, right? How the impulses are moving. An impulse begins when a neuron is stimulated by another neurons or by the environment. As I told you in the start, that impulse can be generated either by the environment or either by the other neuron. And neuron remain in its resting state until it receives a stimulus large enough to start the nerve's impulse, right? The neurons which is present in our body, right, or in any animal body. So these neurons will remain in the rest state till it receive a stimulus, large enough stimulus, so that it can start the nerve's impulse, right? And the stimulus will be from two sources, either from the environment or either from the neighboring neuron. Once it's begun, the impulse travels quickly down the axon away from the cell body toward the axon terminal. So let's suppose the impulse is generated, right? The impulse is generated in one neuron and it is given to another neuron with the help of dendrites. And quickly, this impulse will travel in the axon, right? From one to what? To the axon terminal, away from the cell body toward the axon terminal. It will travel in this way. The neuron cell membrane contains thousands of gated ion channels, right? How it's going to travel? So if you look to the neuron cell membrane, so the neuron cell membrane contains thousands of gated ion channels, right? So the gated ion channels mean that it will be ion channels like for potassium, for sodium, for both potassium and sodium, and for different ions. At the leading edge of an impulse gate, sodium channels open, allowing positively charged ions to flow into the cells. So let's suppose if you look here, just look to this point. Let's suppose impulse is generated, right? So impulse is generated, it will travel in the axon to where it will travel through the axon till the axon terminal, right? And we know that the cell membrane of the neurons contain thousands of gated ion channels. So let's suppose the neuron is stimulated for specific ions, let's suppose for sodium or potassium, right? So it is stimulated for sodium or for potassium, so it is already connected there with what? It is already connected there with the cell membrane. So let's suppose it is stimulated for ions of sodium or sodium ion. So it will open the sodium ion channel which is present in the cell membrane, right? And it will allow the sodium ions to enter into the cell, or also it can occur in the negative side to leave the cell, right? The inside of the membrane temporarily become more positive than the outside. So for this reason, the inside membrane just for a short period of time, temporarily, right? It become more positive than the outside, reversing the resting potential. And thus, when it's become more negative, more positive, so it reverses the resting potential. This reversal of charges from more negatively charged to more positively charged is called Nerv's impulse, right? So when the reversal of charge from more negative charge to more positive charge, it is called Nerv's impulse, or we can say an action potential. It's produced what? It produces action potential. The reverse of what? The reverse of charges from negative charges to more positive charge. This is called action potential, right? What is action potential? Who can tell me what is action potential? Action potential is the reversal of charges from more negatively charged to more positively charged is called action potential or Nerv's impulses. You can see here, right? Now if you look here, so this is positive. If you look here, it become more negative, right? So this is more negative. It's create what? It's create action potential, right? Or we can say Nerv's impulse. All of them are what? All of them are negative, right? But you can see here it creates some positive charge. From a negative, it become temporary positive, right? So when it becomes temporary positive, this is called what? This is called action potential. Once the impulses pass, sodium gate close and the gated potassium channels open, allowing potassium ions to flow out. So once the impulses passes, right, the sodium ions which receive sodium ion from outside, it close, and which channel is opened? The channel which is responsible for the removal or for losing the potassium ions. It will allow the potassium ions to flow out. This restores the resting potential so that the neuron is once again negatively charged on the inside. So when it restores, right, when the potassium ions leave the cell, right, leave the neuron, so it restored the resting potential, and thus the neuron is once again negatively charged, right, on inside. Clear? If you have any doubt in this, you can ask me. It is clear? Okay. So then the last point which we are going to discuss, it's about what? It is about the threshold frequency or threshold. Okay. Not all stimuli are capable of starting an impulse. If we have a stimulus, right? So all the stimulus are not capable of starting impulse. The minimum level of stimulus that required to cause an impulse in neuron is called the threshold, right? Or we can say threshold frequency. So all neurons which is present in our body or which is present in the animal body, so it need a specific stimulus, right? Specific amount of effort is required. If it is sense of touch, if it is sense of smell, if it is sense of sight, vision, hearing, right, taste. So it's required a specific range to stimulate the impulse, to generate the impulse, right? And that specific level or that specific range is called threshold, right? Any stimulus that is weaker than the threshold will not produce an impulse. So if you have a stimulus which is weaker than that given frequency, with that given level, within that given range, so it will not produce stimulus, right? It will not produce impulses. Why? On the other hand, we have what? On the other hand, if the frequency is more, so it cannot also produce impulses, right? Like example, you can take the example of sound, right? Example of sound. So the sound which we are capable for what? The sound which we are capable to hear, the sound which our eardrums can receive. It's ranging from 20 hertz to 20,000 hertz. If a sound is less than 20 hertz, so our sense of hearing, it will not sense it, right? It will not produce impulse. And if it is more than that of the 20,000 hertz, so we cannot hear it. Same, it will occur for all the cases. The brain determines if a stimulus like the touch or pain is stronger or weaker from the frequency of action potential. So the brain determines if a stimulus like touch or pain is stronger or weak for the frequency of action potential, right? The down and the up, right? The low level and the high level, right? The low range and the high range. It will be monitored by what? Or it will be determined by the brain, right? It will be determined by the nervous system itself, mean the brain, right? Like the lower 20 hertz and higher 20,000 hertz. It will be determined or it will be monitored by the brain itself. A weak stimulus might produce 3 or 4 action potential per second, while a stronger one might result in as many as 100 impulses or 100 potential per second. If the stimulus is weak, so it will produce 2 to 3 potential or 2 to 3 impulses per second. But if it is stronger, so it will produce around about 100 potential action potential per second, right? So there is the threshold frequency. Another example, a general example I can give you, that sometime if you eat some sweet thing, right? Like you take tea or coffee, but it's sweet, right? And after that, you are going to, no, just revise it. Let's suppose you take a few cube of sugar. You eat a few cube of sugar, and after that, you are going, after just a minute or 2 minutes, you are going to take a cup of tea, right? So then you are not going to feel the taste of the coffee or tea. It will not feel sweet. Why? Because of the threshold frequency. The test receptors are the test buds which is present on the tongue or in our mouth, right? If we take a cube of sugar, so it means that we increase the threshold frequency. The threshold frequency for the sugar is very high, right? Now the threshold frequency of the test receptors which is present in our mouth, it has been already on the maximum, on the peak, right? And after that, if you are going to take tea, so the receptor already took a high sweetness, so now it will not detect the low sweetness of the tea, right? So you can take this an example. Just take a few cubes of sugar and eat it, and soon after that you can take a cup of tea, so you will not feel the taste of the tea. Because of what? Because of the threshold frequency of the receptors. Clear? Okay. The next point or the last point of this which we are going to take, that is a synapse, right? The synapse. So if you have any type of doubt, any type of question till this place, you can ask me. Now, in simple word, I can tell you what is synapse. The point at which neuron transfer an impulse to another cell is called synapse. Meaning the transfer of signal between two neurons, right? From the axon terminal to the dendritus of another neurons. From the axon terminal of one neuron to the dendrites of the other neuron. Okay. At the end of the neuron, the impulses reach an axon terminal, which may pass the impulses along to another level, another cell, right? Mean that when the impulse is produced, so it will travel from the dendrite toward the cell body, and from the cell body to the axon, and from the axon to the axon terminal, right? And from the axon terminal, from this point, from the axon terminal, it will travel to the next neuron, right? To the other neuron. So now the junction which is between the axon terminal and the dendritus of the next neuron, this transfer which occurred at this point is called what? This is called synapse. A motor neuron, for example, may pass impulses to a muscle cell, causing the muscle to contract. Right? Let's suppose there is a motor neuron, and it is going to pass impulse to the muscle cell to cause contraction and relaxation. Right? So this will be given in the form of synapse. The point at which a neuron transfer an impulse to another cell is called synapse. Right? The point in which a neuron transferred an impulses to another cell, that is called what? That is called a synapse. Clear? Or a space called the synaptic cleft separate the axon terminal from the adjacent cell. There is a place which separates the axon terminal of one neuron to the adjacent cell, mean to the dendritus of the adjacent cell. That space is called synaptic cleft. Right? That is called what? Synaptic cleft. You can see this place, right? This area you can see in diagram. This is called synaptic cleft. Clear? The axon terminal and the synapses contain a tiny vesicle filled with a neurotransmitter, right? Now, if you look to the axon terminal at the synaptic, so it contains a tiny vesicle, small structure which is filled with the neurotransmitter. So, what is a neurotransmitter? These points you can see, right? These yellow dotted structures which you can see. These are called neurotransmitter. Actually, what are the neurotransmitters? Neurotransmitters are actually chemicals, right? That chemicals which are going to transmit an impulses across a synapse to another cell. The chemicals which are responsible for what? The chemicals which are responsible for carrying the impulses from the from the axon terminal of one neuron to the dendritus of another neuron, right? They are called what? They are called neurotransmitters. These are actually chemicals. Okay, so this is all about this lesson. If you have any type of doubt, okay, once they have done their work, the neurotransmitter, right? Once they have done their work, the neurotransmitter are released from the receptors on the cell surface, right? They are then broken down by enzyme in the synaptic cleft or taken up and recycled by the axon terminal. So the function of these are chemical, right? The neurotransmitter are chemical. So when the neurotransmitter ends or finish its function, so these neurotransmitter will be taken by the neurons and it will be destroyed in the synaptic cleft, or it can be destroyed by the enzyme, it can be denatured by the enzyme, and it can be recycled again to the neurotransmitter when there is a new signal or another signal, right? So this was all about today's lesson. If you have any type of doubt, any type of query, so you can ask.